and the title of my article is The Protective Effect of Hyperbaric Oxygen and Ginkgo biloba extract on AB 25 to 35 induced oxidative stress and neuronal apoptosis in rats. Or in simpler terms, a new therapy that forces Alzheimer's to attack itself. So first off, what is Alzheimer's? Alzheimer's is a neurodegenerative disease where plaques and tangles in your brain cause your neurons and synapses to die off in your cerebral cortex, which is the portion of your brain that functions in learning and memory. Alzheimer's is the leading cause of dementia, or is the most common form of dementia. It accounts for almost 50 to 80% of all dementia cases. And in patients with Alzheimer's, your newer memories are often more affected than your older ones, even though patients with Alzheimer's also can have trouble remembering older memories. And as the connections in your brain continue to be lost, your cognitive function worsens, and this can include your learning, your memory, your attention, perceptual skills, and many more. So as you can see in this picture, the brain on the left is a healthy patient, and the brain on the right is a patient with Alzheimer's, and you can see the extreme dynamics leading to the overproduction of ROS, and this leads to something called oxidative stress. So what is oxidative stress? Oxidative stress is an imbalance between these free radicals and your body's ability to neutralize them. And the way your body neutralizes these free radicals are through antioxidants. And what antioxidants do, as you can see here, is they can give electrons to free radicals to stabilize them. So this is where the hyperbaric oxygen and ginkgo come in. Hyperbaric oxygen releases more oxygen into the bloodstream, and that can neutralize these free radicals, as well as increasing the activity of SOD and GSH. And SOD and GSH are enzymes that act as antioxidants in your body. And then ginkgo contains flavonoids, which have also been proven to have antioxidant effects. So they were hoping that these two therapies were able to act as antioxidants and reduce the number of free radicals in the body. Any questions? Okay. So this is the oxidative stress pathway. Like I said before, it starts with these free radicals that can lead to mitochondrial dysfunction, which leads to oxidative damage. This leads to further DNA damage, which then leads to apoptosis, or programmed cell death. And in this case, that's the death of your neurons in your brain. So, kind of to put the whole thing together, oxidative stress is leading to an abundance of ROS. And this ROS is causing your DNA to be damaged, therefore leading to apoptosis and Alzheimer's. But also, this damaged DNA is leading to APP not functioning correctly, which is leading to the AB plaques that I mentioned before, as well as starting the whole oxidative stress cycle again. So what this paper hoped to do was use hyperbaric oxygen and ginkgo to reduce the number of free radicals and therefore lessen the neuronal apoptosis and hopefully have less Alzheimer's in the brain. So the study, they had 6 groups of rats, a normal group, a sham group where they just injected saline, a model group where they injected the rats with the amyloid beta, so that's basically the group of rats with Alzheimer's. Then they treated one group with hyperbaric oxygen, one group with ginkgo, this EGB 761 is just the ginkgo extract, and then a combination of the two therapies. So the way that they conducted the study was with the Morris water maze, and what this is there's 4 quadrants, and it's submerged with water, and there's one platform in quadrant 1 that's 2 centimeters below the water level. So they put the mice in either quadrant 2 or quadrant 3 and gave them 90 seconds to find the platform. Now, the healthy rats should be able to find the platform faster than the rats with Alzheimer's, because the Alzheimer's rats have impaired cognitive function. So the first set of results, escape latency on the left, this is the higher the bar is, the longer it took the rats to find the platform. So basically all of these days show the same results. Just on the 6th day, the results are more pronounced because they've been treated for longer. So as you can see, the normal group, they were able to find the platform much quicker than the rats with Alzheimer's. But then if you look in the combined treatment group, they were able to also find the platform a lot faster than the rats with Alzheimer's that weren't treated. So this shows that their memory and concentration was actually increased due to the treatment. And then on the right, island entries, this shows how many times the rats actually were able to just swim into quadrant 1. So even if they couldn't find exactly where the platform was, at least they could remember what were free radicals in your body. So you want more of these. So again, you see that both the treatments on their own had protective effects, but again, the combined treatment was more effective and had significant difference compared to the model group. And then MDA. MDA is a marker for oxidative stress. So as you can see in the model group, the levels of oxidative stress are much higher in rats with Alzheimer's, and then with the treatment, the levels of oxidative stress came much further down. Okay, so then they wanted to look at the effects of these two therapies on apoptosis-related proteins in specific. So if you look at BAX, BCL2, and cytochrome C in specific, BAX and cytochrome, these three proteins are produced in your mitochondria. So the imbalance of mitochondrial dynamics that I mentioned earlier would also lead to the imbalance of these proteins in patients with Alzheimer's. So BAX and cytochrome C are proteins that induce apoptosis, so you want lower levels of these. And you can see that in the model groups, both cytochrome C and BAX, there were higher levels of them, and the treatment groups were able to bring levels of these proteins down. And then inversely, BCL2 is a protein that inhibits apoptosis, so you want more BCL2, and again, you can see the positive effects of these treatments on the right. And then finally, just culminating all of these, actually showing less neuronal apoptosis in the rats' brains. These dots, all of these dots are neurons, and over here you can see they kind of look exploded. So those are neurons that went through apoptosis. So this is the rats with Alzheimer's and no treatment, and this is the rats with both combined treatments. So you can see the extreme difference in levels of neuronal apoptosis, and then this graph just reinforces that idea. Yeah?
Are any of those the treatments alone, hyperbaric oxygen separated from?
This is just the oxygen, and this is the ginkgo.
Was there a significant difference between D and F?
D and F, no, I don't think so. You can see down here the hyperbaric versus the combined. Okay, so now what? So hyperbaric oxygen and ginkgo proved to be positive therapies for Alzheimer's, and this is important because they didn't just attack the symptoms of the disease, they actually prevented the neuronal apoptosis that causes Alzheimer's. And also, oxidative stress is relatively new in the scientific community, so this wasn't a big step for just Alzheimer's, but also for the whole scientific community. And now researchers are kind of looking to other diseases and if oxidative stress could possibly lead to other diseases as well, because right now these are just a few of the effects of Graff and oxidative stress on your body. So I know you've probably all heard that blueberries and things like that have antioxidants in them. So just kind of reinforcing that idea that we really do need to take care of our bodies because oxidative stress can be very harmful to ourselves. So I said before, free radicals are always present in our body. They're produced through energy-producing reactions in our cells. So if I have free radicals in my body, will I get Alzheimer's? The short answer is no. Oxidative stress isn't the only precursor to Alzheimer's, but this paper took a big step in showing that oxidative stress definitely plays a big role in it and could have a big link to possibly curing Alzheimer's in the future. So